You guys so much for being here for today's episode of a lunch a la carte, which is a lunch and learn that is sponsored by the Cart Fund. It's an opportunity for our donors, our volunteers, our supporters, and the general public to learn more about what's going on in the field of research for Alzheimer's and dementia. We appreciate you being here today, either live or watching our recorded version. Just a couple of quick announcements. I want to make sure that everyone is aware and already have it on your calendar to join us for this year's annual meeting of the CART Fund. It will be July 30th and 31st of 2026 in Charlotte, North Carolina. Registration is not open yet, but it will be before too much longer. But we want you to go ahead and save the date to join us for a reception on Thursday evening with our researchers who are selected this year, and then an all-day ceremony and seminar on Friday where we get an opportunity to hear from some of those researchers and learn more about the projects they're working on. We had a record number of applicants this year for our grants. For the first time ever, our previous record was 93 applicants. This year, 272 applicants for grants from the CART Fund. Those are in the process now of being narrowed down to our top 15 finalists. And I'm pleased to announce that our executive committee is looking at awarding another record amount of grants this year. As you may recall, in 2025, we awarded a record of $2 million in one year. And this year, our goal is $4 million. So stay tuned for more information on that. It's my pleasure to ask our past board president, Bill Parker, to join us now to introduce today's speakers.
Thank you very much. Dr. Kim and Dr. Quinn, we record these sessions and make them available to all of our Rotarians, and we direct the Rotarians to where the recordings are so that they have an opportunity to hear and study and everything. So we appreciate your input. And with that, I'd like to introduce Dr. Luisa Quinti, who received her undergraduate degree in chemistry from the University of Pavia in Italy, and then pursued a PhD in chemistry at the Manchester Metropolitan University in the United Kingdom. She moved to Boston, Massachusetts as a postdoctoral fellow and started working at Massachusetts General Hospital as a biochemist. Dr. Quinti is currently an instructor in the Department of Neurology, working with Professor Rudolph Tanzi. She has been actively involved in the development of therapeutics for several neurodegenerative diseases since 2005 and is passionate about developing and implementing reliable models of Alzheimer's disease that can identify novel drug targets and fast track the search for a cure. Together with Professor Yeon Kim, she is translating novel technologies developed in the lab into robust drug screening platforms to find Alzheimer's disease drug candidates that can be readily evaluated for human trials. Now, Dr. Kim has been studying pathogenic mechanisms of Alzheimer's disease for more than 16 years. He and his team investigated physiological and pathological functions of BACE1 and presenilin gamma secretase, two key enzymes that regulate generation of amyloid beta, a major pathogenic molecule associated with Alzheimer's disease. Dr. Kim and his colleagues characterized four novel presenilin gamma secretase substrate proteins and BACE1 substrate proteins that explain how BACE1 regulates neuronal activities under physiological conditions. Excessive amounts of BACE can disrupt the neuronal activity, making nerve cells more vulnerable to aberrant firing and perhaps seizures, which may explain the higher risk of seizures in Alzheimer's patients. And this is really important. Recently, Dr. Kim's lab, together with Dr. Rudy Tanzi's lab, developed an innovative three-dimensional human stem cell culture model of Alzheimer's disease. By cultivating genetically modified human neural stem cells in a 3D gel system, they were able to recapitulate key events of Alzheimer's disease pathology, including amyloid beta plaques and the tau tangles, for the very first time. In other words, their lab model can produce what starts Alzheimer's disease and can be observed. This unique human neural cell culture model can be used for large-scale, high-throughput screening of novel therapeutic targets, which is not feasible in our current Alzheimer's disease mouse models. And mouse models are used by almost all of our researchers to date when doing drug testing. Kim's Alzheimer in a Dish model has been listed as one of the 10 breakthrough technologies of 2015 by MIT Technology Review. And it also won the 2015 Smithsonian American Ingenuity Award. In 2022, Drs. Quinti and Kim received a $300,000 CART grant. And this CART grant was to explore current malarial drugs and see if they could be used for eliminating the amyloid beta plaque buildup in the brain, so that we have a drug that is considerably cheaper than the drug that has recently been approved by the FDA, which costs hundreds of thousands of dollars for the treatment and is beyond the scope of any average patient. So with that, Dr. Quinti and Dr. Kim, the floor is yours, and thank you so much for everything you do to try and find a cure or a prevention of Alzheimer's disease.
Hey, thank you very much for nice introduction. I'm going to share my screen quickly, and then can you see my screen? So yeah, I think it's a very nice introduction. I really appreciate you introduced for so much details, and I think you already cover everything, I would say. So yeah, my name is Yeon Kim, and Associate Professor of Neurology at Mass General Hospital and Harvard Medical School. And I'm the primary PI of this CAST funding. And this is Luisa Quinti, doctor, and she is also working within the same institute. She's a co-PI, and she's working on drug screening part. So I'm working on the model part, and she's working on drug discovery. And before starting, I would like to express our really great appreciation for the generous support from CART and its members. And due to the federal funding uncertainty nowadays, and these private funding and foundation funding is getting more and more important to keeping these critical human health researches. Thank you very much. So in the first part of my talk, I'll introduce our 3D cultural models, not only basic concept 3D cultural model, but I want to show you the ones just recent our progress, which is human brain organoid models of Alzheimer's in a dish. So now previous models just have a cells growing in the 3D gel dimension. Now this model it's really have a human brain. It's really having activity, everything. And then we are developing Alzheimer in this brain organoid model. And I think there's a lot of advantage here. We are really hoping this new model replacing mouse model. And I'll explain in the presentation why we are not really big fan of mouse model at this moment. And in the second part, Luisa Quinti will show how these advanced model can be used to find the drugs for Alzheimer's disease. And also, she's going to show how we combine these models, screening, and bioinformatics, also computational AI-based bioinformatics can be combined together and figure out which is going to work for the humans in the end. And that's actually one of the things. And of course, in the last part of her talk is about progress in anti-malaria drug. This anti-malaria drug is one of the hits from this screening. So she's going to show that the new mechanistic idea, how we found these are working for Alzheimer's patient brains. And that. Okay, let's go to the next. So I think I didn't need a lot of introduction of Alzheimer's disease. As you know, now in the United States, 7.2 million patients, actually a lot of number, and 39 million in worldwide. And this is because of our getting into super aged society now. That means Alzheimer's aging is a key risk factor. And now there are more than 20% within the 65, that means aged society. That means Alzheimer's incidence is getting higher and higher and expected getting more. And if you're looking at right side, the table graphs, this is the change of death rate of 5 major cause of disease in United States, and their changes in the last 10 years. And as you can see that among these, among these 6 major cause of disease, there's a lot of improvement. Like for example, HIV, you see minus 42%. That's actually a huge decrease of death rate by the drugs and others. But unfortunately, if you are looking at Alzheimer's, it's 68% increases. Among these major 6, only Alzheimer's, we are having huge increases every year. So I think that's really big problem we need to solve at this moment. And this is exactly what Bill just mentioned. The 99.6% of Alzheimer's drug candidates failed in human clinical trial. These are the graphs showing that currently in the pipeline, but we know very well there's a lot of problem in these going into human trial. And why the main question I have and Rudy has is why the drugs very effective in mouse models and some other models are all failing in the human trial. That's actually one of the questions we had in almost 10 years before. And this is what I got it from the Rudy's. And I think our conclusion is very simple. Mouse is not human. And mouse may not have Alzheimer's in the end. So we always call it mouses might be the most fortunate species in the whole world because scientists fix the almost 100,000 of disease, at least in the mice. So if you are a mouse, you have a drug for every disease. That's actually what's happening. The problem is that, is it really true that these mice represent human conditions clearly? And that's actually one of the questions we had. So, and go back to here, let's briefly show that amyloid plaques and neurofibrillary tangles. These 2 things are needed to understand my talk. So these are, you can see that Alzheimer's patient brain versus normal brains. And 2 peculiar pathological marker is amyloid plaques in outside of brain cells. And neurofibrillary tangles is inside the neuron. These aggregates, you can see it in the patient. And going to the details, these amyloid plaques generate from very tiny small peptides, so-called amyloid beta. And they're aggregating in disease conditions and make these big aggregates. And for the neurofibrillary tangle, it's made out of protein named tau. Tau is protein generally working in the microtubules in the neuronal cells, but in Alzheimer, somehow they are dissociated and aggregated. And these 2 things are very peculiar in Alzheimer's patient brain, not in other diseases. So out of the years, almost like 2 decades, these beta amyloid cascade hypothesis, that's actually framework of how we understand Alzheimer's. So this idea is that the amyloid beta, which is pathogenic molecules, accumulates and then aggregates. And these aggregation trigger these vicious cascade into synaptic neuronal loss, gliosis, and hyperphosphorus tau tangles, and eventually getting into neurodegeneration because brain cells are dying. And we so-called this is ATN framework. So amyloid, tau, neurodegeneration. The problem is that mouse model fail to recapitulate this cascade. So mouse model, you hear about it, they do show amyloid beta plaque with the genetic modification, and they do show gliosis, and then they do show the memory dysfunction. However, they never develop neurofibrillary tangle nor neurodegeneration by amyloid. This is one of the biggest issues because if you have a drugs working for amyloid in mouse model, yes, it actually makes sense. But if you have a drugs blocking neurodegeneration or even later stage of tangles, these models are not going to show you that whether it is working or not. So that's one of the biggest problems. And that's why we studied our 3D culture model. And probably you are the first one showing that if we are using human neuronal cells and others, unlike mouse, we can develop A beta, tau tangles, and neurodegeneration. That's our 2014 paper that we got award and others. And I can briefly show these. I think is I don't think I need to go get to detail. This is cellular model we generated at the time. We genetically modify human stem cells, make a new brain cells, and grow them in three-dimensional gels, like Bill said. And then that leads to what we surprisingly not only A beta aggregation, but unlike mouse model, they can develop A beta induced tau tangle cascade. That was very exciting to us. And one of the idea here, I can just go to very fast. So the reason why we use 3D culture model is very simple. If we are using 2D culture model like others, you can see that A beta is not aggregating, just diluted out every time we change the media. But if you are looking at 3D culture, what we found is that these gels is working like brain matrices. So we are making small brains in a dish wells, and then that actually make them amyloid accumulates, aggregates, and trigger these vicious cascade. That's our concept of 3D culture model of Alzheimer's in report in 2014. And I can just show this. This is one shown in the New York Times, 2014. And these red, really big red one is the amyloid plaque, which is the first ever generated in the human cellular models. And it's a three-dimensional structure. You can see these are going up and down, and those are generated. And that's one of the very big achievement. And another one, so I'm going to go very fast here because that I want to give Luisa a lot of time. So and not only these aggregates, if you're looking at right side, you see these very scary looking filamentous structure. These are the really inside structure of amyloid beta plaques. So other cellular model, you may have A beta increases, but in our model, they are making these filamentous vicious structures inside the culture, and that's actually another achievement, our 3D culture model. And this is tangles, and then I can just go to the right side, EM. Again, these tangles are not just the tau protein aggregates. They are making unique structure, so-called PHF, paired helical filaments. And then what you can see in the bottom of the right panel, you see these helical helix, and then you can see turnover every 80 nanometers. And that's the peculiar structure character of PHF tau tangles. You can only see it in the Alzheimer's patient brains. And ours are only recapitulating at that moment, still identified. So you can see that these models are for the first time, unlike mouse, and recapitulate very robust human-like Alzheimer's patient pathologies in a dish for the first time. And I'm going to skip here. So I think this is actually interesting. I know this is too much, but many Alzheimer's genetics is not really saying Alzheimer's is happening by just increase of A beta peptide. If you are looking at that mutations, everything, what they do is sudden change of pathogenic A beta 40 versus 42 is really key for these pathogenic cascade. And the reason I'm showing you this is that this is how you can make either good A beta 40 or bad A beta 42. So it's almost like when you wake up in the morning from the bed, and we stand up in the right side or left side. Depending on that, you're getting really all the great A beta 40 benign peptide. But if it's right side, you are getting into A beta 42, which is causing Alzheimer's disease, aggregates in your brains. So this is actually the genetic idea, and we generated models, and that's at the 2020, and we dissected it. Basically, what this model does is that if you are enriching 42 cascade, you are getting plaques and tangles. But if you're enriching opposite cascade, you don't get any plaques and tangles. And this is very well controlled experiment in our view, because previous models just you have a control and Alzheimer's. And there's a big problem because there are too many other things going on. But here you can see that there are models that only increasing A beta 42, not 40, is the key for it's really recapturing Alzheimer's pathology in a dish models. Okay, how much time I have? Pardon? Okay. So yeah, we can go, and then I'll show you these brain organoid model. So previous model, I also show you 2014 and others. It's all great, but except they are not really brains. They don't have brain structure, brain activity, nothing. Just they do have neuronal cells growing in the three-dimensional culture. So next, our challenge is, can you make a more Alzheimer dish model, 3D model, but more brain-like structure, brain-like activities and others, and we turn into brain organoid model. The different from 3D culture brain organoid, a brain organoid human stem cells, and they are making developmental brains. So they make a structure. I'll show you how it is going there. So I'm gonna go here. So we genetically engineers, exact same like 3D culture model. One of them is having pathogenic 40, the other 42, and the other non-pathogenic 40. And we generated them into the human stem cells. And these pathogenic A beta will be expressed upon doxycycline treatment, which is we are making Alzheimer's after these brains get developed. Actually, that's very exciting. And then you'll see these kind of things. And I'll show you one of example. So you see really nice picture. This is a replica of what's happening inside the dishes. And then this is very similar to your the cortical plate structure in your brain. So this is like, these are not just like 3D culture. They are making these development structure, almost like brain-like connections. And that's what these does it. And then what we do is that we make this system expresses A beta 40, 42, and others, and making Alzheimer in a dish after different. And this is one of the things I can see. I hope you take a look in the bottom middle, and you see the really bright spots. And this is so-called April green fluorescence spots, and this is amyloid plaques. And as you can see is that we cannot see anything in the control. We cannot see in the A beta 40, only A beta 42. You can see these a lot of spots. This is what's really happening similar to Alzheimer's patient brains, and that's aggregates of A beta. And we go into the tau tangle pathology. Again, this is EM studies. So you can see bottom of the pictures, and you can, again, so this is PHF, tau filamental structure, recapitulated in the brain model for the first time. And this is really exciting for us because these are much more complicated, sophisticated model, even have some activity, and then we encapsulated Alzheimer in a dish model again. So this is very exciting. And another biggest excitement to me is that we also found this model can recapitulate not only A amyloid, tau, but neurodegeneration. This is quite robust. Basically, what you can see here is that these organoids, not everything just in the A beta 42 overexpression, they are showing robust neurodegeneration. You can see these neurons are almost gone. It's black means there's nothing left. And you can see these are shrinked. And we think this is very similar to what you can see in the Alzheimer's patient. I show in the main picture that in the later stage of Alzheimer's patient brains are shrinked. And that's what we see in our organoid model. Organoids are shrinking because cells are dying a lot. And there's a lot of cells that are dying here. You can see that the only A beta 42 neurons are dying first, and later astrocytes go forward. So altogether, it's very interesting because we didn't see it in the previous. Any questions? So we didn't see in the previous 3D culture model because those are not really recapitulating this brain structure. Now, not only A beta and tau tangles, we are looking at neurodegeneration recapitulated. So it's very exciting. And this is one of the things, I think this is really striking picture for us. So what's happening is that we look at in the electron microscope and looking at these dying cells, Alzheimer model dying cells directly into it. And then what you can see here is that these are the normal cells, these are the nucleus, and there's the mitochondria. In the middle, what we see is that you already see the nucleus is getting distorting. And another big thing is that you see a lot of these big fractures. This is so-called the granular vascular neurodegeneration bodies, which is known for the Alzheimer's patient. And then you can see these, a lot of fractures generated here, not in the control. Go further, and this is a big enlarged picture. So these vectors, you can see these white or nothing there, just very small spots. These are the kind of digesting contents inside. Only these things are left over. But another biggest surprise to me is that you can look at these, the yellow arrowed ones, and these balloon-like structure is mitochondria. These mitochondria is supposed to be like very nice structure in the left side, but what you see is that these Alzheimer model and these mitochondria become swollen and become like a balls. And we check out the old publications, and we are very surprised to see that indeed there are reports about these swollen mitochondria in Alzheimer patient brain. So what we are looking at is very similar to what you see in the Alzheimer patient brain, which is not recapitulated in mouse models or even previous model. So this is very interesting, but the most exciting thing is that now we are targeting neurodegeneration because right now there's ATN. So we have A beta accumulation, tau tangles, and then there's a neurodegeneration. And all the drugs targeting right now is amyloid. And that's the drug Bill and I just discussed. But there's no drugs targeting neurodegeneration at this moment because there's no neurodegeneration models. But now we have it. So this is one of the exciting findings we had. So you can see that what we found is the drugs that reversing mitochondrial deficit in our model. And not only they are just blocking mitochondrial damages, but what you see is that they are completely reversing neurodegeneration. Despite there's a lot of A beta still accumulating, but with this particular drug, these neuronal cells are surviving without any damages in our experiment system. So to me, is that this is another way to think about Alzheimer drugs. Not only we are looking for the drugs blocking A beta accumulation, but these drugs can block A beta induced neurodegeneration. That's actually one of the ideas we find in this model. And these are our conclusion in here, right? What we see is that these models are recapitulating for the first time ATN framework, amyloid, tau, and neurodegeneration for the first time. And we're very excited. This is going to give us a new opportunity to find a new drug, which was cannot be doable with the previous model. And that's what we are developing right now. So, and before moving into Luisa drug screening, I can just show you, this is how we are looking at we want this model to be drug screening, but previous organoid is kind of very tough to grow them in the dish and others. And this is so-called like a high throughput culture format. So you see these 96 plates and these tiny plate, you can see these very small dots in the middle, right? That's an all brain organoid. Almost like there are 96 Alzheimer patients in a single small plate. And we can treat drugs and everything in these individual organoid. And this is going to give us the idea how we can fix all the damage from the A beta accumulation and others. And we're quite excited whether these models can be used for the testing drugs, not only mouse models, but these brain organoid in a dish model can be used to test it before going into the clinical trial and others. And that's actually what I have for today. And Luisa is going to take over, and then she's going to give you all the things. And that's the major things.
So I'm just going to talk about the drug discovery part, so the drug screening. So here there is a comparison of different models, the mouse model, the organoids, and the 3D culture model. And of course, going from the mouse model to the 3D model, we lose complexity because, of course, a mouse has a lot of more complexity compared to a 3D culture model. However, we increase reproducibility, and also we lower the time that it takes to screen drugs and the cost. So for a mouse model, we think that to develop AD, it takes about 12 months. And then you have to do the treatment and the analysis. So if you use 8 mice per compound, it costs about $500 per compound. Instead, if we go to the organoid model that group is developing right now, that is progressing very fast, but still is an expensive model because it costs about $150 per organoid to grow them for about 12 weeks. And it takes 5 to 12 weeks to do the testing. Instead, if we go to the standard 3D culture model that was developed in 2015, we can use 4 wells, so 4 replicates for each drug. and it cost about $5 per drug. And in 6 weeks, we start and finish our culture, and there are about 2 weeks of analysis. So it takes 2-3 months for each experiment. And we have been doing this since 2015. So initially, we started the screening in collaboration with Dr. Yeon and Dr. Wong at the Houston Medical Methodist Hospital. We screened a big library of over 2,500 compounds, which included FDA-approved and biologically relevant compounds. And the cell line that was used was developed by Dr. Kim's group, and the cultures were done for 6 weeks. And we were looking at compounds that were reducing pathogenic tau, so phosphorylated tau. And we have some images of these in C. And the readout was microscopy, so high content analysis. So the reduction of tau was considered the drug to be a hit. And we found 38 primary hits that then came back to our lab, and we started retesting them at multiple concentration and to assess that they were not toxic. And we kind of prioritized them and tested a few recently in collaboration with another group in a different human model, which actually starts from a D patient fibroblast, which is considered a late onset Alzheimer's disease model, and found that there is a matching between our hits. So this is another representation of the big screening that we did at the beginning, where we found the 38 hits. So as you can see, each dot that you have represents one compound. We found a series of compounds that are decreasing phospho-tau, but also some compounds which are increasing phospho-tau. And these are the ones that we would like to know, because they're basically over time, they may give people the disease. And then we divided them into the mechanism of action, found out what these compounds are doing, and the group that contains most of the compounds is the neuronal ion channel. And then all the others are grouped, and then we more robustly tested them. So we looked at phospho-tau over tau and found out that actually 8 of these compounds are robustly doing in other similar model. And also we look at what happens at A beta. So we looked at the ratio A beta 42/40 and saw if the compound are only modulating tau or A beta.
So maybe I can kick in that we have 8 finalist drug we think is very exciting and blocking there. And the reason we are just blurring is that because we are currently preparing manuscript. We're trying to make sure that it's published, everybody see what is the really hits in our model. Because it's not always that these drugs are directly useful for the drugs, but to me it's that it's very important to disclose information to the older people as soon as possible. Who knows, maybe some of them out of these hits can be the future Alzheimer's drugs. Right.
And after doing the FDA-approved and biologically relevant compound, we also looked at natural products and look again for drugs with our 3D-AD model that were reducing phospho-tau over taus. Why? Because there are advantages and disadvantages of natural product. In general, natural products are mixed of reagents, so that can be a disadvantage, but they tend to be less toxic. And also they are more expensive can be more expensive to make, but on the other hand, more readily available in nature. So we screened the library of 800 compounds, and they were again grouped with different mechanism of action. And out of these natural products, we found the anti-malaria drugs. And these are the 5 compounds.
Maybe I can kick in. Sure. So out of these, the natural product is exciting is we have at least 12 dozens of the hits. And many of them is that actually found it's used for the health supplements. They are not drugs, but they've been having consumed by human by the supplement of these other things. So that's another idea we had. Maybe not only FDA approved drugs, but the natural product compound or chemical compound has been widely used for health supplement for different purpose, and maybe use it because they are not toxic. They are okay to consume by human without any huge amount of clearance, right? Go ahead.
So we were very excited when we found this anti-malaria drug because it's a family of compounds which are structurally very similar, and they all have this endoperoxide bridge, which is this oxygen-oxygen bridge, which is expected to be responsible for their anti-malaria property. The starting molecule is artemisinin, with which a Nobel Prize was awarded in 2015 to Dr. Yu. And then another important molecule is DHA, the second one, because this is the active metabolite of the artemisinin family. And the others are present in the WHO list of drugs. They've been developed for different purposes and given for anti-malaria resistance. When anti-malaria resistance occurs, a different molecule gets given to, yeah.
So I think it's one of the things we want to clarify is that anti-malaria drugs, there are different types. So what we found is not all the anti-malaria drug is working. In this particular chemical groups, only artemisinin groups are the ones working. And then the amazing thing to us is that all of these modified version works almost same efficacy in our 3D culture model of Alzheimer's. So that means they are sharing some common mechanistic mechanism. That's why we are working on it. But it's not like other anti-malaria drug, there are several famous ones. Those are not the ones working. It's very toxic in our space.
And it is not known in details the mechanism of action of the artemisinin with the anti-malaria drugs. So it's what is known is that artemisinin will bind to the heme of the bugs and form an activated complex, and this will alkylate many proteins inside the parasite, causing death. And then this is a little bit more in detail saying what happens, that you're going to have many toxic species which will cause the death of the bug, which are the alkylated proteins, carbon radicals, and reactive oxygen species, which will result in the death of the bug. And now we have started screening these anti-malaria drugs in our model. And the important proteins for Alzheimer's disease are effect on tau and effect on A-beta. So the panel on the left shows that all of the screen artemisinin compounds are reducing phospho-tau over tau, so the pathological tau, and two of them do it with statistical significance. And then we also looked at what happens to insoluble A-beta and insoluble A-beta 42. Insoluble A-beta is increased, while there are no changes to insoluble A-beta 42, and the pathological A-beta is A-beta 42. Also, we did some mechanistic aggregation studies because A-beta in Alzheimer's brain is aggregated. So we have a very simple assay which looks at the aggregation of A-beta 42 or A-beta 40 in vitro in the presence of the drug, as measured by thioflavin T fluorescence. We follow this aggregation for 4 hours, and this what happens. So in blue, we have what happens without the drug. So the control, the protein will aggregate over time, but the aggregation is much faster in a much higher rate in the presence of the anti-malaria drug. And with the anti-malaria drug, we also see a dose effect. So increasing the amount of drug, there is more aggregation. So 10 micromolar in green versus 100 micromolar in red. And when we don't have the oxygen-oxygen bridge, so these 2-deoxyartemisinin, the aggregation is reduced. So that points to the fact that these endoperoxide bridge is important for aggregation purposes, interaction with A-beta. These are other electron microscopy studies, say to the one that Dr. Kim showed before. And we have what happens with the drug in the presence and with and without RT ether. So in the top, we have the control. There is these filament forming and aggregates. And with the RT ether, there is no change in terms of type of fibrils. While in a beta 42, in the presence of RT ether, there are no longer fibrils. So there are remains, but they are not structured. So they will not be able to aggregate. And I went one faster. And these are what happens with the other two of the drugs. The top is artemisinin, and the bottom is deoxyartemisinin that does not have the two oxygen. As you can see in the case of deoxyartemisinin, you can still see the filament. So again, the endoxopyroid bridge is important for interaction with a beta 42.
So I think is that this is one of the starting point we didn't know it before getting the funding and these all the studies, how the antimalarial drug intercalate and changes amyloid beta aggregation and toxicity. And our working hypothesis now is that it seems like they are almost like similar to they are targeting antimalarial box, but looks like they're also targeting these pathogenic A beta aggregates in the brain, and they are binding and then kind of destroying the structure. That's a kind of like a very amusing thinking, because it has the same mechanistic idea, but in the blood, they're killing box. In the brain, they are destroying these pathogenic A beta aggregates, not the conventional way. It's more like more aggregation, right? Okay.
And the last piece that we're going to show is transcriptomic approaches. So I wish I had a little bit more time, but basically we treated our 3D culture with the drug, extracted RNA, and sending for analysis, looking at the gene, which are the differences in gene between the control and the drugs. And so this is the first step when you do principal component analysis. Then you look at how much the genes are changed versus the significance, and you form this volcano plot. And I'm going to show the one for our drugs later. And later on, you do look at the genes that belong to certain pathway, which pathway do the drugs have in common, and they're modifying. So these are the results for our anti-malaria drugs with the volcano plot. These are called volcano plot. On the right side, there are the genes which are upregulated, and on the left side, the genes which are downregulated. And we did it for the all four anti-malaria drug, artemether, artemisinin, dihydroartemisinin, artesunate. And then we looked at the pathways since we have the gene. So these are how much our drug changed the canonical pathways. And as you can see, we have 4 drugs which are very similar. So they have a lot of pathway in the same color, while RT ether is the one that gives as a kind of reverse mechanism. Instead of upregulating, which is shown in orange, it's downregulating. So it's in blue. And also, we looked here at which one at the top most significantly upregulated 15 pathways for the drugs which are in red, in orange on the left. So we have artemisinin, DHA, artemether, and artesunate. And this one points out that, this analysis points out that our artemisinin family is reversing the stress-induced translational deficits, which we know is a critical pathogenic cascade in our 3D AD model. So that's how we think that the drug are working, and that's how they could work for AD, and that's what we are studying further.
So I think this is one of the great advantage we got CART funding, because these huge amount of data sets has been global with the CART support, and these data will be deposited in the public domain. Everybody can see it. This is probably first ever data on mice looking at how anti-malaria drugs are working in Alzheimer model human neurons and how they change these all the changes. Of course, we have working hypothesis I show you. We are going forward and probably have a publication soon. But whole thing is that we are also depositing these all the data sets, and that will be very helpful resource for the future studies and also medical discovery, right? Okay.
So then I'm just going to finish that. Therefore, we have studied, we looked at what did anti-malaria drugs do on A beta and tau. We also look what they do on aggregation, and we are now taking advantage of the result that we got with the transcriptopin approaches to understand further what is the mechanism. And just as a disclaimer that our data do not mean that the anti-malaria drugs are the cure for AD. So most of the anti-malaria drugs actually do not enter the brain. So the other problem is that the chronic administration of the anti-malaria drug has not been proven to be safe, because typically anti-malaria drugs are given for a few weeks every time there is the infection, but they are not given for 10 years continuously. So extra precaution needs to be taken for an elderly population.
So the instance is it that probably this is the answer for your question, Bill. So instead of taking this malaria drug as the Alzheimer cures, what we propose is that these all the information, how they are working in our model is critical to find new drug targets. So what we are looking for is doing something very similar to this anti-malaria drug does based on mechanistic studies. But instead of directly injecting malaria drug into the brain, it's better to find alternatives doing. And that's why we did all the work, because we know all the detailed mechanism now. And what we are looking for is something mimicking same results, but still brain permeable. That's actually what we are looking for right now. Mm-hmm.
So this is a very crowded slide, but so the two currently approved drugs are for Alzheimer's disease are Leqembi and Kisunla, and they're both targeting A beta oligomers. However, there are a lot more parts of the AD pathology where we can intervene. We can block in A beta production, aggregation. We can work on tau. We can also look at boost microglia so they will phagocytose better A beta, or once it's activated, microglia releases cytokines. So we can stop the release of cytokines because that also causes neurodegeneration. We can further look down at blocking like apoptosis or necrosis. So in the words of Rudy Tenzi, our head of the unit, he says that if we just stop at treating plaques, it's like once you have a fire in a forest, you are turning off the match. So it's not enough. People already have plaques, probably have intervened to something that will block other mechanisms as well as the plaques.
So I think this is one of the understanding now. As you can see, this is quite complicated structure. And then in the old times, we thought that amyloid beta stays all the way down to the end. And then when you get rid of amyloid, we are hoping that everything is reversed back. But as you can see, amyloid is more working like triggers or match fire. And once the forest fires everywhere, just blowing up matches doesn't really reversing everything there. And that's why we need additional drug candidate combination nowadays, instead of just happy that we reduce amyloid. I think that amyloid is a starting point. What we did now is that additional drugs that can be used together or independent use for these late stages, right?
And this is our acknowledgement to our unit, the whole of DU's lab, postdoc, instructor,
Yeah.
and then my team, they do drug screening, microglia and 3D, and then the person that did most of the electron microscopy, which is Yeon Kim, and then our collaborator at Beth Israel, Du didn't have time to go through this paper that was recently published. And the collaborators are Houston Methodist Hospital, which they started with us the drug screening of the FDA-approved compounds, and also the paper that we're about to publish. So with them. And then for funding, the NIH and CART. And we are ready to take a question,
So
if there are any.
I think one of the things is that we really rushed everything because we hope we have a time to get questions. But I feel bad that we prepared too many slides today, so I had to skip many things. I'm so sorry for that one. So we are done, right?
Yeah.
Go ahead. You're muted. Bill, you're, yeah.
I'll tell you, I keep looking at this stuff. I keep seeing that the level of complexity just keeps advancing. Every time we think we're down to a level where we finally gotten to the root of the matter, there's a level of complexity times 10. And it is just amazing how far we've come since we issued our first CART grant in 1999, when we were looking at LR11 or APOE 3 and 4. But what really knocked my socks off on this for the two of you is the fact that you now have a model in a dish that you can actually start the process, because the real issue we've had in researchers in the past is what starts the avalanche, what pebble on the cliffside starts all the rocks rolling down. And I think you are finally getting to the point where you now can at least see what's causing it, what's starting it, so that we can start looking for interventions to prevent, to pause, and possibly reverse. And that to me was the thing that really opened my eyes in your presentation. Plus the fact that I took anti-malarial pills when I was in Vietnam for 2 years. So I'm going to be safe, I think. With that, I'll turn it back to you.
Does anybody have any questions? Well, this has just been fabulous. Thank you guys so much. We're so glad we were finally able to get you guys on with us. We appreciate all the work that you are doing. And of course, we will broadcast this to our YouTube channel afterwards. A few little edits, we'll get that up as soon as possible and we'll share that. But again, as always, we appreciate your support of CART. We appreciate what you're doing for the cause, and we definitely appreciate you all taking time to be with us today.
Thank you very much. And